Good afternoon, everyone. I'm Steve Yoswick. I'm the Senior Science Writer for TGen. And before we start, I want to mention that we will be recording this event, and that recording will be posted as soon as possible and no later than 4 today at www.tgen.org/news. Again, www.tgen.org/news. You'll open up the first item on that web page, which is today's press release, and you'll find a link to the recording at the top of the page as soon as we post it there. Again, we'll do that by 4 p.m. So for TV reporters, please feel free to record this event directly from your computer. And following the introductions, if you have any questions for today's panelists, please post them to the chat box, which you'll find, I believe it's down at the bottom of your screen there. I think we already have some chats coming in. And let's see here. Okay, so we are again recording, and for those who just came on, yes, we are recording. No one else will be able to record this, but we'll post that recording to the tgen.org/news webpage. All right, so I believe Paul and all of our panelists, go ahead and unmute yourself if you would.
Hey, thanks, Steve. I feel like I know at least 90% of the people on this call already, but I'm Paul Keim. I'm a Regents Professor at Northern Arizona University. And myself and Michael Warraby from U of A and Dave Ingelthaler from TGen, we got together, put our heads together back in, I think it was February, and we decided that Arizona needed to have the best science tackling this pandemic, the best science that could be available in the entire world, and we need to have those scientists focusing upon the problems here in Arizona. So we formed something called the Arizona COVID Genomics Union, and we are a loosely affiliated, informally affiliated cooperative group of scientists from the University of Arizona, from Arizona State University, TGen, and from Northern Arizona University. So we've been tackling the genomic side of this pandemic from an Arizona-centric standpoint for over about 6 months now. And the press release that's coming out today is in fact announcing one of our peer-reviewed papers on the analysis of the early pandemic in Arizona. This paper is appearing in MBio, which is one of the flagship journals of the American Society of Microbiology. And it is what we call a peer-reviewed paper. So this paper has already been looked at by other scientists who have approved our methods and approved our conclusions. The two reviewers are not anonymous in this case. One is from the university, from Oxford University in the United Kingdom, and the other is a bioinformaticist, geneticist from the Centers for Disease Control in Atlanta. So this paper really represents, the best science possible when it comes to using genomics for understanding the pandemic, and it's based again right here in Arizona. Not only is it doing fantastic science for the state of Arizona, but it's setting a model for how you do this type of analysis to groups around the world. So the fact that it's peer-reviewed and it was done in such a rigorous fashion means it's going to have a great impact, not only in Arizona, but everywhere. Today, we thought we'd do a press conference in a slightly different way. Normally, you see people like me or Mike Warraby or Dave Ingelthaler here. But in fact, there's a whole team of people behind us who are actually doing the work. And so we're going to highlight 5 early career scientists who are a part of this work. And we're going to let them tell you about the work. And we're going to let you then ask them questions about the work. So those 5 early career scientists are Jason Ladner from Northern Arizona University and the Pathogen and Microbiome Institute. Nice wave there, Jason. Brendan Larson from the Ecology and Evolution Department at the University of Arizona. Jolene Bowers from the Pathogen Genomics Division at TGen, located at TGen North in Flagstaff. Crystal Hepp from NAU and the Pathogen and Microbiome Institute. And finally, Ephraim Lim from the Biodesign Institute at Arizona State University. So what we're going to let them do is each of them give a little bit of their perspective on this study and tell you a little bit about what they did and how they view this before we open it up for questions. So we're going to start with Dr. Jason Ladner, assistant professor from NAU. Jason.
Yeah. Hi, everyone. Thanks, Paul. So I just wanted to kind of set the stage for our discussion of this manuscript by talking a little bit about just how exciting it is right now in terms of the use of genomic data generally for studying infectious disease outbreaks. And the reason for that is just because of various advancements in genome sequencing technologies that now really allow for routine, near real-time sequencing of viral genomes during these outbreaks. And because, as I'm sure you've heard about, these viruses evolve pretty quickly through the accumulation of mutations, we can actually use these genomes to track the spread of viruses through space and time. And therefore, these viral genome sequencing efforts can really help to inform the public health responses that are associated with these outbreaks. And in fact, the ongoing COVID-19 pandemic has really clearly demonstrated the incredible capacity that we now have for rapidly generating these viral genome sequences directly from clinical samples. So I checked yesterday, and on the online database, GIS-AID, that's being used to collect these genomes from around the world, over 100,000 SARS coronavirus 2 genome sequences have already been uploaded and publicly released for analysis and interpretation of what's happening during this outbreak, which is really unprecedented and really exciting. Now, as Paul mentioned, we're here to talk about the first paper that the union has put together, which really, I think, lays the foundation for the continued work that this group is doing. Specifically in this paper, we're reporting SARS coronavirus 2 genomes that were sequenced from 84 different patients. All of these patients were tested in Arizona and collected mostly during March, a little bit during early April of this year. And so this is really giving us a snapshot of the very early portion of the SARS coronavirus 2 outbreak in Arizona. And by comparing these genomes with those that were generated from around the world, it wasn't 100,000 at the time that we were doing this analysis, but it was still a very large number. We were able to characterize the dynamics of viral spread during the first few months of the outbreak in Arizona. And I think this really nicely sets the stage for similar analyses that we're working on now that are focused on the peak period of transmission in the state. And I'm going to let my co-authors tell you a little bit more about the specific analyses and findings from this paper.
Thanks, Jason. So next up, we're going to hear from Brendan Larson, who's a doctoral candidate at the University of Arizona. Brendan.
Yeah, so Jason mentioned one of the unique ways we can use these data is to look at differences, little differences between viral genomes to determine where these specific introductions came from. And by sequencing these known introductions, we can also determine if sustained outbreaks occurred from these very specific introductions. So for example, the very first case of COVID-19 in Arizona was a student who was returning back to Maricopa County from Wuhan on January 19th. And this person reported to the clinic very quickly, and their genome was sequenced by the CDC, and we used it in our analysis. This patient isolated for 14 days, recovered fully. There was no evidence of any transmission events from this patient. And by comparing their genome with all of the other genomes that we sequenced, we were able to determine that public health interventions do work. And in fact, we did not detect any viruses that descended from that first initial introduction. And so by testing, tracing, and isolating, public health people were able to make sure that no more infections descended from this first introduction. So that's one of the powers of this approach of genomic epidemiology, is that we can determine which methods work which methods don't work for controlling the virus. And with that, I'll turn it over to the next person.
Yeah, Brendan, great example of how we worked with public health and everybody else in this field. Next up, we're going to hear from Dr. Jolene Bowers, assistant professor at TGen. Jolene, tell us about what you were doing on this whole thing.
Yeah, hi everybody, thanks, and I'm really happy to be representing TGen in the ACGU. And as many of you know, we are a research institute actually, and even though we're studying infectious agents that are important in medicine and public health, we really were not testing patient samples. But in February and into March, we really decided to change gears and go for it. And that's, you know, because with all of the people and the knowledge and the experience that we have at TGen, we really were perfectly suited to respond to this pandemic. And our response really coincided nicely with these early pandemic days of Arizona that we write about in the paper. And so we started designing the assays and doing development work to be able to test patient samples right around the time that we, the ACGU, determined that the virus was actually being introduced into Arizona that was going to result in widespread transmission. So in mid to late February. And then we actually started testing, we continued to build our lab out, and then we were able to start testing actual patient samples in early to mid-March, which is right around the time that the community transmission within Arizona was being recognized and reported on. So our timing was great to be able to get some of these early Arizona cases so that the ACGU could really tell the story of the early pandemic days of Arizona. And so TGen North now, we're continuing to do the genome sequencing and helping to analyze the data with the ACGU, and we're now the reference lab for all of Arizona for SARS-CoV-2 sequencing. We're up to about 1,000 genomes a week. We're aiming to get to 10,000 total genomes, which will really give us, give Arizona the most complete statewide genome map, if you will, really anywhere.
Yeah, great, Jolene. I mean, you really highlight the fact that if we hadn't jumped into this early, I mean, we could go back and we can go and look at the pandemic now, but because of this paper and those early efforts of TGen, we now have the basis and foundation to move forward and understand what has happened over the summer and into the fall. All right, so now let's jump over to Dr. Crystal Hepp. Crystal is an assistant professor from the Pathogen and Microbiome Institute at Northern Arizona. Crystal.
Hey, thanks, Paul. I just wanted to cover briefly one of the main methods that we used in this paper to produce a lot of our results, and that is the molecular clock analysis. So one advantage to working with RNA viruses, such as SARS-CoV-2, is that they evolve at a relatively constant rate over time. So this means that they accumulate mutations at a steady pace, and this accumulation has often been compared to the ticking of a clock, and so we call this the molecular clock method. So SARS-CoV-2 in particular gains about 1 mutation every 2 weeks, and knowing this molecular clock rate allows us to effectively count up the number of mutations in a particular data set over time so that we can back estimate when a particular lineage entered a location, just like we've done for how separate lineages have entered Arizona. And so this analysis indicated that even though the first case was identified in January and the vast majority of our samples were collected in March, we found that the majority of importations from this particular data set occurred in February. So that's what I wanted to add. Thanks.
Thanks, Crystal. Mutating viruses sounds pretty scary. I think we need to talk about that some more in a moment. But next up, I want to turn the microphone over to Ephraim Lim, who's an assistant professor at Arizona Biodesign at Arizona State University. Ephraim.
Thanks, Paul. Hi everyone, so my name is Ephraim Lim. I'm assistant professor at the Biodesign Institute. And I think this study is terrific because it highlights a collaborative effort by multiple research institutions across Arizona State. And that's a very unique part of this study. What we have here, as Paul highlighted, was representation from TGen, from Northern Arizona University, from University of Arizona, and as well as from us from Arizona State University. So at ASU, as you're probably aware, we have a lot of research efforts going on right now with COVID-19, and this is one of those efforts. So we have been sequencing the SARS-CoV-2 genome since early in the pandemic. That was through our surveillance studies at the Arizona State University Health Services. But then it became really clear quickly that this is a bigger problem than just us, and that we really need to work together as a team. And so this was a natural fit to work together with the ACGU. And as Jason has pointed out, this is not the end, this is just the start. We have still a lot more work going on to sequence and understand the viruses of SARS-CoV-2 in Arizona State. However, the upside is, I think that compared to where we were in March and April, I think we now have a very good handle of the SARS-CoV-2 isolates that are currently in our community right now. And this gives us a very good starting point from here on out.
All right, Ephraim. So I'm not getting any questions in the chat box, Steve. Are you getting questions?
Yes, sorry, if you have any questions, please go ahead and click the chat button at the bottom of the screen, and there'll be a chat box over on the right-hand side where you can go ahead and write your questions.
I can certainly start off with questions here. You know, in the paper, you know, mutating viruses, that's a really scary thing. At least it sounds really scary. So I'm going to maybe the first question should be about a particular mutation that we did observe in these that's been widely reported. It's a mutation in the spike protein that changes one amino acid to another. I don't know, would one of the panelists like to discuss the G614d mutation, explain what it is and why it might be important? Somebody jump in there or I'll point at you.
Okay. I could say something, a brief introduction. So, I mean, this is, as journalists, I'm guessing you've probably heard about this mutation if you're covering the outbreak, but this is a mutation that occurs in what's called the spike glycoprotein. So this is a viral protein that's embedded into the envelope of the viral particles. And so these are proteins that are all over covering the outside of the viral particles. And in fact, it's these particular proteins that sort of in the electron microscope images give these viruses the appearance of crowns, and that's where the name corona comes from. And so the important thing, there's a couple of important things about this protein in general. One is that because it's on the outside of the viral particle, it's one of the primary viral proteins that our immune system recognizes and responds to. And two, it's the protein that directly interacts with proteins on the surface of our cells in order to facilitate entry of the virus's genome into the cells. And so what a group of scientists, I think at multiple institutions, recognized was that there's a particular mutation, a change from a D amino acid to a G amino acid. So that's why it's D614G, occurs at position 614 in the spike glycoprotein. And that this mutation seems to be sort of taking over in most of the outbreaks around the world, meaning that most of the cases that are currently occurring in most places are of viruses that now have the G residue at this position as opposed to the D residue. And we did, in the data that we have, sort of explore what's been happening with this mutation in Arizona. The Arizona outbreak in general was actually already dominated by the G variant very early on, but at the same time, we did see that the relative proportion of the genomes that we sequenced that were this G variant did increase over time, which is consistent with this idea that's coming out that this virus may be able to transmit more efficiently than the D variant of the version of this virus. Although I would caution that that doesn't necessarily mean anything about the disease caused by this virus. So it doesn't mean that this G variant is more pathogenic or more likely to be associated with death or severe impacts. We don't know anything about that yet, and from what I understand, what has been shown does not correlate with that.
Okay, Paul, we're starting to get some questions in on the chat, and I'll go ahead and read the first question here, and you can select somebody who you think might be best to answer this. The question is there evidence of super spreader events early on, or do the data point to a particular date or date range during which there was a lot of spread of a particular mutation?
Anyone want to tackle that? Super spreaders in Arizona? Brendan, you look like you've got something to say about that.
No, well, I was trying to think. I don't think our data can show necessarily if there were super spreader events. So that would be more boots on the ground epidemiology where they do contact tracing. Since this genome evolves fairly slowly, it's difficult to make inferences about events that occur within a 2-week time frame. So I don't think our data necessarily shows anything about super spreading. I don't know if anybody else has anything.
Crystal.
Just to add, so we don't have data about super spreader events at this point in time, but one thing that we are doing, expanding on, is looking at individual lineages and how their population sizes have increased and decreased over time. And so that can tell us how well a particular lineage has persisted or increased in magnitude in the Arizona population, but at this point in time, we don't have those data.
Yeah, you know, I might just say that our data is early pandemic, and so we're really catching the first little bit of this. And so the super spreader events, which definitely occurred, we know from the traditional epidemiology that these super spreaders have occurred, and that those are really going to have the impact on the pandemic that comes after this particular study. And so with this early analysis, we should be able to point to the roots of that and then how it takes off. Good question.
One more thing, just to piggyback on that, Paul, a little bit is, I think that one thing that's changed a lot over the course of the pandemic, not necessarily that we see from our data exactly, but is the amount of movement that people are doing around the country and around the world. And so what we see in the early data that we've been looking at and been published on is that you actually see a lot of instances of completely identical viral genome sequences present on multiple different continents. And what this speaks to is the fact that people were traveling all over the world at high relative frequencies and moving this virus around quite a bit. And what I expect is that we start to dig more deeply into the later samples, because there's less movement of people right now, that we're going to see that a lot of these sequences are sort of based more locally in Arizona or in other places, and we'll be able to track those movement events more easily.
Yeah, nice.
Okay, so here's another question, and maybe this is one Crystal might want to follow up on based on her earlier comments, but what is the significance of the mutation rate? Is this a slow mutation rate or a relatively fast one that we're seeing in this virus?
I think that Jason Ladner can also probably answer this question as well, but because he and I work on a variety of different viruses, so I've worked on West Nile virus, St. Louis encephalitis virus, those viruses that are transmitted in mosquitoes, HIV in the past, and in comparison, this virus is relatively has a slower mutation rate than these other viruses do.
Yeah, I'll just add, so for an RNA virus, meaning a virus that has a genome that's composed of RNA, this virus and coronaviruses in general have a relatively slow mutation rate. And that's because coronaviruses have some capacity for correcting errors that are made by polymerases. So polymerases are the enzymes that copy nucleic acids. Turns out that all polymerases are error prone. Viral polymerases, our own polymerases that are copying our genomes, but we have a lot of mechanisms for error correction. So when the polymerase makes those errors, we have proteins that are able to recognize those and repair those. And that's why, so relative to our own mutation rate and the mutation rates of other cellular organisms, this virus still has a pretty fast mutation rate. But relative to other RNA viruses, it's somewhat slow.
But I guess in terms of what it really means, in terms of significance, I guess if it's mutating like once every 2 weeks or so, I guess I've been told by some researchers that we may already be past the point where there's going to be a universally applicable vaccine. But I guess how much time would go, like how much time would need to go by before this is getting to a level where there's too many different lineages or maybe too many different mutations, or where we're getting, how much time would need to go by before we see another really significant mutation?
Yeah, Ephraim, you want to tackle the vaccine? I think this really plays the vaccine versus the mutation in the targets.
Sure, that's a good question, Amanda. So yes, I think what Jason and Crystal are mentioning is the overall mutational rate, and that's relatively slow. However, as we found, and others have found as well, that there can be very large mutations, deletions in the genomes of these viruses. These seem to be rare, but can occur. When you're talking about the response to vaccines, though, most of the targets for the vaccine candidates right now are targeting the spike protein. There has been some mutations, but overall, spike is still pretty well conserved. Secondly, in terms of selection, we think about whether mutations might affect the virus being selected for. Right now, there isn't a strong selection in the population targeting specifically the spike protein yet, because there aren't any vaccines out right now. So actually, I can understand why people might speculate that there might be multiple forms or strains and so forth, but based on the evolution of the virus, based on what we're seeing in terms of sequences, actually, I do not expect that there is any selective pressure on the virus right now that might affect the vaccine candidates in the pipeline right now.
Yeah, but I guess the question is like more, how long do we have to kind of figure out this vaccine before we risk a higher likelihood of a significant mutation?
That's a good question. So based on what Jason and Krista are saying, the basic evolutionary rate for baseline of this virus is pretty slow. So based on number one of that basic evolutionary rate, it's probably not going to be too fast. Secondly, what I mentioned is the virus evolves also based on selection. And since there isn't a vaccine right now exerting a selective pressure on the virus population, that doesn't speed up the selection process right now. Does that make sense?
Would that change once a vaccine comes out? Would there be kind of some pressure to maybe increase the rate of mutation?
That's a good question, and probably Jason can answer that better. But based on our knowledge of other viruses, so like flu is a good example of that. We have a flu vaccine every year. And the reason why we have to keep updating our flu vaccines year to year is because of selection. And one of the major drivers of selection in flu is the seasonal flu vaccine itself. Jason?
Yeah, I was just going to add, I mean, I think I was reading something recently that said that basically if you go back to the very first virus that we think jumped, the strain that jumped into humans, and then you take any other virus circulating right now, you're talking about around 10 differences between that initial virus and the virus that you see now. And that's 10 differences across 30,000 nucleotides. And these, at the nucleotide level, these are occurring relatively randomly, right? And so a lot of these actually won't even change the protein sequence. The amino acid level, they'll be what we call synonymous substitutions. And so I think at the sort of thinking about this at the functional protein level, we're still seeing pretty low levels of diversity circulating. And so it doesn't mean that there aren't some variants out there that might be able to escape certain immune responses, but what the frequency of that is in the population and whether it'll be meaningful against the vaccine, I think are open questions. And I don't think we'll be able to give you an answer of sort of how long we have before that becomes a significant problem.
Just to add also, Amanda, to your point about, or your question about if the mutation rate is going to increase over time due to selective pressures, mutation rates are stable over time. What changes is which mutations are selected for and end up rising in the population. So we won't see the mutation rate increase, but we might see different variants different strains of the virus that are rising in the population that we don't see right now. And that could be perpetuated by a vaccine.
Yeah, and Amanda, I might just add that, you know, the Arizona COVID Genome Union is here watching for that very thing. I mean, it was one of the main things that we do is we monitor the mutations, we map those out. We use them for the molecular clock estimates, but as the vaccine rolls out, we're still going to be here. We're still going to be watching this virus. And so the good news is that if something starts to change, we can help direct the altered response to those changes.
Hey, can I actually follow up with maybe, well, a comment first, and then actually maybe a question for my own colleagues. So this is really kind of the first time that we've had a pandemic, a new novel pandemic, where we've had the technology to be able to sample and sequence a whole bunch of representative strains. And, you know, like Jason said earlier, there's already over 100,000 different strains and genomes in the database. And so, you know, that I wonder, you know, could that be feeding into this idea that there's a really high mutation rate, but that's just because we're sampling the heck out of this virus, which is unprecedented. We've never done this amount of testing before for any infectious disease among the population, nor have we sequenced this many genomes so quickly. So we might be seeing, right, would you guys agree, we might be seeing a lot more mutations than we typically would with less sampling, right? Like we normally have been in the past.
I think, I don't want to get too technical, but I think this kind of brings up this difference between a mutation rate and a substitution rate. And so the mutation rate is constant over time. That's the number of errors that the polymerase actually makes in each time that the viral genome is being replicated. What we're actually estimating in our analysis is called a substitution rate. And so there we're actually looking at the number of these mutations that actually kind of go to fixation in the population. Although in this case, the population is kind of the population present within a given host, within a given human host. And so one of the things that does happen, Jolene, and that people have shown is that when you're sampling a bunch of things over a short period of time, it does seem like the estimated substitution rate is a little bit elevated from where it would be if you sampled over a much longer period of time. And we kind of say that this is incomplete purifying selection. So you're sampling a bunch of these variants that are actually have lower fitness overall than a lot of these other variants, and they're going to be lost from the outbreak as you move forward. And so if you just looked over this larger time span, you would actually measure a little bit of a slower relative rate. But my guess, and this is a non-expert opinion on this because this is not something that I study, but I think it has most to do with the behavior of individuals. That the big spike in cases and seeing those plots showing Arizona as #1 in the world per capita finally got through to people that they need to wear masks, that they need to be social distancing. And I think that's why we've seen the big drop in cases.
Yeah, I would add one thing. I totally agree. Definitely, I think finally we're learning our lessons here. That these public health response efforts really do work. But the one thing that I would add to is that we now know quite a bit more about how to do, and there's more people and more resources available to do this contact tracing. And I've seen at least one study that has shown that this rapid response and contact tracing really does save lives. it really does have an impact on the morbidity and mortality of the disease. And some Arizona communities, well, many Arizona communities now have had the luxury of time to prepare before the virus has hit their specific region or community. So that's really helped them to prepare and get the boots on the ground ready to go for these contact tracing efforts.
And I'll add also, this is, I guess, anecdotal at this point in time, but if you were to look at the Coconino County dashboard and look to see when the number of cases started going down in Flagstaff, you will see that those cases started going down when the mask mandate was put into place.
Okay, so I have another question, and that is, what do you see going forward in your work with the Arizona COVID-19 Genomics Union? What would you like to accomplish?
Well, I guess I'll start out just because I'm more on the front end of putting the sequence data out to the ACGU. We're about to bombard them with a lot of the latest case sequencing data. And I think that will really help us to continue the story that we started and talk a little bit more about the mid and these more recent phases of the pandemic here. And also, you know, what this will allow us to do is really start parsing apart some of the more local level stories. And we've been starting to work with the team, and then with, we have really good relations with a lot of public health officials at the local level, county, state, and even federal level with the CDC. And we're able to now start matching up a lot of their boots on the ground epi with our genomic epi data to really start to complete some of those stories.
Yeah, I think one of the big picture sort of questions moving forward that we want to try to address is thinking about sort of the major drivers of the outbreak here in Arizona. And so one of the questions is how much was that big peak in cases driven by sustained local transmission occurring in Arizona, and how much of it was being driven by repeated introductions of the virus from outside of Arizona. And that's something that, especially with this large number of genomes that are available from throughout the country and throughout the world, is something that, along with the genomes that TGen, University of Arizona, and ASU are generating, I think we can start to really parse out.
Also, one idea that we have is if we look before versus after the shelter in place, we'd like to understand how impactful that shelter in place was at limiting transmission across even different counties. So the hypothesis going into it is that when we were sheltering in place, the transmission would have occurred primarily within county. But then after shelter in place was lifted, what we would expect to see is that there was a lot more transmission across the different counties, and then also across different states into Arizona as well.
Okay, so absent a vaccine for the time being, what are the most important things that the public should keep in mind about helping to stop the spread of this disease?
Well, I guess I'll start. The first thing I would say is, you know, exactly what our public health officials and government officials are saying, and that is definitely to mask up. It definitely does help. Practice the social distancing, avoid the big crowds if you can, and if you get together, being outdoors helps. I'm sure, yeah. Do you guys have anything to add to that?
I mean, I think one of the open questions is, sorry, Crystal, is this idea of immunity and what immunity looks like and what's the potential for reinfection. And I think right now, I would urge people to continue to be cautious, even if you have already gotten a positive test or know that you've been exposed. It's definitely been shown recently, although I don't think it's going to be very common. It's been shown that it is possible for people to become reinfected. This is actually, I think, one of the kind of, I don't know if I would call it a successful story, but one of the really important contributions that genomic sequencing has actually made to this understanding this epidemic is that it gave us very clear evidence that reinfections have occurred. We still don't know too much about those reinfections, but I would say kind of forget the idea of these immunity passports and that everyone should continue to follow the social distancing guidelines.
And I think also that we should think of the decrease in the number of cases since our peak as a massive success for the state of Arizona. And now that we've been masking up for so long, it's understandable that participation enthusiasm may have waned or is starting to wane over time. But we have to remember that this is a success and that for us to keep our caseload lower until we get, hopefully until we get a vaccine or yes, until we get a vaccine, that masking up is just really important for us to keep doing.
Okay, so just a couple more questions then. When people have been reinfected, are they being infected with a different strain or the same strain of the virus?
Yeah, I can answer that a little bit. I mean, I think part of it is to think a little bit about what is being asked. Because my guess is the meaning behind the question is the person fully immune to the first version of the virus they were infected with, and they were only able to be reinfected because it's a new version that was not able to be successfully targeted by the immune system? That, I would say, is probably not what's happening. Like we talked about before, the overall level of divergence in the genomes of these different virus variants that are circulating is quite low. So strictly sort of speaking, yes, they're infected with a different variant because the virus is always changing. The probability that they'd be reinfected at a later point in time with exactly the same virus sequence is very, very low. But are they infected with a new variant because they're only immune to the first variant? No, I don't think that's the case.
I would just add one other thing to that too. I agree with what Jason just described, but also point out that people can actually test positive for weeks after contracting the virus. And so sometimes, you know, people might think that there's reinfection happening when it's actually not. It's just this low level persistence of the virus or the viral material, maybe not viable, in the person.
Okay, Jolene, you mentioned that you're ramping up to do 1,000 sequences a week, and with a goal of, was it 10,000 sequences total? And approximately how many sequences have we done so far? I think the last I heard it was about 3,000.
Yep. Yeah, I'd say, you know, we definitely have to distinguish between successful genomes and ones that we've attempted to sequence. So that right now it's a little hard to quantify exactly how many successful genomes we have. But yeah, we're bordering right along around the 3,000 mark for 3,000 genomes. And of course, as our experience is getting, you know, deeper and deeper into this, and as you know, the vendors and supply makers are honing their kits and reagents and things specifically for this, we're getting better at it. And so, yeah, we're almost seeing like an exponential increase in the number of genomes that we're sequencing now.
What do you mean by successful genome?
Yeah, sure. Well, so in some of the cases, you know, people might be infected with a really low level of the virus. And in that case, it's a little bit harder to pull out and target just the virus in that sample to be able to pull it out and get the entire genome sequenced. Whereas, you know, someone that's very heavily infected, they've got tons of virus in that nasopharyngeal swab that was collected. It's a lot easier to pull out that virus to be able to sequence the whole genome. In the cases where we're not successful, what I mean by that is we'll only get part of the genome, or maybe you know, 80% of the genome, but we really want the whole thing, or at least close to the whole thing, to be able to really do these robust analyses with the ACGU.
So basically, you guys are, I just want to make sure I'm getting this right, sequencing about 1,000 a week, but the sample size right now is only about 3,000 like complete genomes.
Yeah.
Okay, thank you.
Yeah.
How often is it that you guys can't pull out the genome, I guess?
That, you know, that's really kind of unpredictable because it really depends on the sample set that we're testing. And so, for example, you know, we might be testing a lot of specimens that are collected from people that are asymptomatic. Say, you know, there's one of our public health officials is screening some population, doing a lot of contact tracing where people aren't necessarily heavily infected, where it's making them sick. In that case, you know, we might get a low success rate, but typically, you know, if we're able to detect the virus and detect it well, then we're successful at getting the genomes out.
Sorry, another follow-up, I guess, just thinking about this from like a common public understanding, if you saw that and you read this as a story, I feel like the conclusion that a lot of people would jump to is, oh, well, if most of the ones they are successfully doing are this one strain, like, do you think that the asymptomatic ones are just as likely to be that strain, or like the ones that you aren't able to sequence? Or I don't know.
Yeah, I think I get your question, sure. And it really depends on the context. And, you know, one thing to keep in mind too is the people that are most heavily infected are the ones that are the most infectious. And so they're the ones that are going to be spreading it. So, you know, for these people that aren't that heavily infected and where we're not as successful at pulling out the whole genome, we're not missing very much because those probably aren't all that meaningful epidemiologically, if that makes sense. Go ahead.
To keep in mind, though, is that the sort of viral load in the sample, and therefore the ability to easily sequence a genome, is also going to depend on the timing of the sample during infection. And so, you know, different people are going to end up being tested at different stages of the infection. And so it doesn't mean necessarily, you could have two individuals infected with exactly the same virus, and there are a lot of different variables that could tell you whether we're going to be able to easily sequence it or not. Could be when during the infection was the person sampled. It could have to do with the person's own immune genotype. Could have to do with what viruses that person was previously exposed to. So there's a lot of different variants that can sort of impact the ability to get a large amount of viral nucleic acids that's going to be totally independent of the actual genotype or sequence of the virus.
I think, in other words, to summarize all that, I wouldn't read too much into an unsuccessful sequencing of the virus genome. As Jason has pointed out, there might be very simple reasons why we can't pull up the whole sequence. It doesn't mean that they're not infected or we're not confident that they're infected. No.
Okay, so here's another question, and whoever wants to take this, jump right in. when you're looking at the global genome database, have you seen any evidence of Arizona exporting the virus and sending outbreaks elsewhere?
It's a good question. I would say that in the data set that we've published thus far, we have not had the resolution to determine that with any kind of real certainty. We also, the analysis that we've done most thoroughly and that we've published is from a time in the outbreak where the number of cases occurring in Arizona was quite small compared to the number of cases occurring in many other regions around the world. I think with the data set that we have sort of moving forward, I mean, I'm sure that it has happened. Whether how much those other outbreaks that have been seeded, sort of how many cases have occurred as a result of those, I don't know, but I wouldn't be surprised if moving forward we see some pretty strong evidence to support those types of export events.
Okay, and Jolene, question for you. How long do you think it'll take you to get to 10,000 sequenced viruses?
I am hoping by probably within the next month, we'll see that. Yeah.
Okay. Okay, so are your genomes representative of the infections as a whole? And I'm guessing this question means the genomes that you're seeing, the strains that you're seeing, are they representative, you know, of what you're seeing elsewhere around the world or around the United States?
I'd say in the very early part of the outbreak that we've published on thus far, they're relatively representative, meaning that we see many, if not most, of the major lineages that were circulating around the world at that time, also evidence of those strains being present in Arizona. I think that's also something that's likely to change as we move forward.
How so?
Yeah, just to clarify the question, I guess I'm wondering if it's sort of a representative sample of the infections within Arizona. Are your findings, do you think that they reflect the whole of what's going on, or is there a blind spot out there of genomes that you're not sequencing for whatever reason based on where you're getting your samples to sequence them?
Yeah, I think it's a good question. It's something we've started to explore more quantitatively recently, sort of how well are we getting a representative sample of places. So basically, how well does our genome set correspond to the number of cases that have been reported? My understanding is that we have a few geographical blind spots because of certain counties sending their samples to a certain testing facility, which is not currently sharing those samples with us for sequencing. I think Jolene could respond a little bit more to that, but that is my understanding. There are sort of a few counties where that might be the case, but overall, we've been getting a pretty good representation of at least, I'd say 12 of the Arizona counties.
Yep, you're right. The one thing that I would mention is we are being sent samples, positive case samples for sequencing from the state lab, and that is pretty representative of the Arizona statewide. However, yeah, there might be some gaps in there, excuse me. But we are also starting to get some positive case samples from other reference labs. And we're waiting on the data to be able to tell where these samples are from. sometimes our sequencing efforts are faster than the filling in the metadata that go along with the sample that it comes with. But as we're working with our public health partners a little bit more, we'll be able to fill in that story a lot better, and yeah, as time goes by. And I know, Crystal, not to put you on the spot, but I know that some of your analyses, like Jason was saying, have they can actually give you an idea, right, of how well representative a sample set is of what's actually out there.
Well, one thing that we are able to do when we count up the number of mutations that's present in a particular data set at a particular time, we can look to see, okay, there are this many cases in this particular population, and we're looking at the genomes from this particular population. And if we see that there are more mutations than expected, then we can assume that we are undersampling people in that particular area. And also, if we are oversampling the people and there's only a few genomes there's only a few genomes that are have been sequenced, we know that we need to increase our sequencing efforts in that particular area. And then I'd also add that, of course, results are going to be inherently biased by the availability of testing in a particular area, and also by testing blitzes that different organizations might be putting on. So, for example, Northern Arizona University is putting on a testing blitz right now. So in Coconino County, some of the results might be biased by the number of NAU affiliated individuals. So we have to take those biases into consideration when we are trying to think about Arizona as a whole.
Okay, so on this session, we looks like we have about 7 more minutes. I wanted to talk a little bit more about the paper that was published, and about the very first case that we saw here in Arizona. Does this become a model or a representative way that you wish all of these cases might have been handled? And is it still a good goal to try and look at it that way going forward, even though there's so much infection out there?
Yeah, so I think that this is not the only example of something like that. I heard a talk from some folks in Wisconsin at the University of Wisconsin in Madison recently, where they described a very similar situation where they had one early case documented towards the end of January that led to no other downstream infections that were detected. And so, yes, I mean, this is the ideal scenario when the level of infection is low and every single case can be the priority of sort of making sure that we prevent that person from passing the infection on to someone new. But we kind of have to be realistic when we're seeing much more widespread transmission as far as what's possible in terms of contact tracing and prevention for every individual case.
Okay, getting you to brag just a little bit, how do you think your efforts with the union compare to other efforts that you've seen around the nation and across the world?
Jason, I'm going to defer to you.
I would say that globally, the effort has been really incredible. Paul would probably do a better spokesman job of saying how special we are, but I actually think that the global effort focused on characterizing this virus at the genomic level, as well as in other levels, is really incredible. And so we see a ton of sequencing pouring in from all over the world. Last time I checked, I think the UK is kind of kicking our butts a little bit as far as the US in general. Like, they've really been on top of it, and I think we probably still need to step up our game a little bit, but I think we're doing pretty well.
Jason, did you say that there were 100,000 genomes in GIS AID?
More than 100,000, yeah.
Okay. Well, so if I don't think that the current genomes that are coming out of TGen are all uploaded at this point in time, but when you think about, okay, there are 3,000 genomes and there's been just over 100,000 that have been submitted, it seems like Arizona's effort is pretty fantastic for just one particular state, I'd say.
Agreed.
And it's not just that. We have the whole of Arizona state covered. There is not one state in the United States that can make that claim. There are very specific studies in New York, very specific studies in Washington state, very specific studies in Idaho and so forth. But what we have done here is we put together all our resources and expertise to focus on the state level. And I think that's the unique part of this.
Okay, just a couple of additional follow-up questions here. Do we have data on how shelter in place impacted the spread? And I think, Christopher, I think it was you who mentioned earlier that we're going to be looking more in depth on that. But is there anything that we can tell from what we've seen so far?
You said as how children are impacting the spread?
Do we have data on how shelter in place impacted the spread?
Sorry, shelter in place. No, that's something that we plan on looking at right away. What we really want to do is compare the phylogeny, so the relationship of all of the strains before shelter in place to the relationship of all the strains after shelter in place. And during shelter in place, we would expect that we're going to have county-specific lineages, and after shelter in place was lifted, we expect that there's going to be a mishmash of all of these different counties mixing together. But yes, so that study is underway.
Okay, we still have just a little bit of time. Are there any other questions that anybody would like to ask?
Yeah, so a couple questions. Are we going to be looking for reinfections in Arizona or using the genomics to study or kind of like watch out for potential reinfections and confirm those cases?
So understanding reinfections would mean that we would have to have access to very personalized data. And at that point, so at this point in time, we're not looking at the level of a particular individual. So I think at this point we're not, but Jason, do you have anything to add to that, or Ephraim?
I would say it's going to depend more on the public health agencies to be tracking at that level and to identify cases that are potentially reinfections. And then in that case, absolutely, if they send us the samples, we would sequence them, characterize the both strains. You have to make sure you have good quality samples from both infections in order to be able to do the comparison. But if that type of metadata has been tracked and they can send us those samples, we would absolutely do the sequencing to support that.
And on our side, yes, we have those planned as well. We're working with our clinical partners, so Mayo Clinic, Abrazo Health Hospitals, where there are a lot of clinical studies going on like that. And this longitudinal question is a really important one, and we are tracking those. Okay,
so we have less than a minute, and I think I'd just like to go ahead and close out now if we can.
Actually, just one last.
Go ahead.
One question. Somebody said that the proportion of that mutated spike strain grew over time. What are the percentages of how it grew over time, like initial percentage versus now?
Ooh, had that up recently. I would guesstimate that it started out at about maybe 30% and has gotten up to close to 95%. Oh, sorry. Let me say that again. Started out at about 70%, the G variant, and then has gotten up to about 95%.
Okay, so Jason, Brendan, Jolene, who's off now, Crystal,